Your gut knew before your brain did. The receipt is time-stamped under 100 milliseconds. In 2018, a team at Duke University, led by Diego Bohórquez and his colleague Melanie Kaelberer, published a paper in the journal Science that quietly broke 100 years of physiology. They had identified a class of cells in the lining of the intestine. They called them neuropod cells, and these cells form actual synapses with the vagus nerve. Fire 1 and glutamate hits the brainstem in less than 1/10 of a second. That's faster than a blink, faster than the conscious thought of being afraid. The gut is wired into your skull the way an eye is. For decades, the textbook story was that your gut talks to your brain slowly through hormones drifting through the bloodstream. A sluggish, chemical telegram. Bohórquez's lab proved the line is direct and the line is fast. Every healthy adult on the planet has a sub-second channel running between their intestinal wall and the same brainstem region that processes everything your body has to say. By the end of this, you'll know exactly what your gut does in the first seconds of fear, the first minutes, and the first hours. You'll know which signals you can feel and which ones never reach consciousness at all. You'll know why a system perfectly designed for a predator on the savannah is the same system that now keeps you awake at 3 in the morning over an email you haven't sent. The answer reaches back 500 million years. And by the time we get to the modern half of it, you're going to recognize yourself in the broken parts. So let's go through it carefully. Start with the wire. The vagus nerve runs from the base of your skull down through your chest and into the upper 2/3 of your gut. You've probably been told it runs the rest and digest system. What you weren't told is the direction of traffic. Roughly 80% of the fibers in the vagus nerve are afferent. Afferent means the signal travels from the body up to the brain. Only about 20% carry commands from the brain down to the gut. 4 out of 5 lanes on that highway are pointed up. Your brain spends most of its day listening to your stomach. The cell bodies of those upward fibers sit in 2 small clusters outside the base of your skull, called the nodose ganglia. From there, the axons run down into the gut wall, where they sense stretch, chemistry, temperature, and the presence of bacterial fragments. They relay everything up to a brainstem region called the nucleus of the solitary tract. From that one relay station, the signal fans out to the hypothalamus, to the locus coeruleus, which runs your alertness, to the central nucleus of the amygdala. The amygdala is the part of your brain people associate with fear. So the anatomy is screaming the thesis. There is a fast, dedicated, hardwired path from the inside of your intestine to the part of your brain that decides whether to be afraid. In 2022, a team led by Wenfei Han ran the cleanest demonstration of this anyone has managed. Working in rats, they used chemogenetics to selectively activate the vagal afferents coming out of the gut, and the animals became measurably more anxious. They selectively lesioned those same afferents, and the animals became chronically less anxious. That's not a correlation. That's a causal anatomical pathway from gut to fear circuit, demonstrated in mice. The same vagal architecture is conserved across mammals, including us. So the wiring almost certainly works the same way in a human skull. We just can't open one up to prove it directly. Bohórquez's contribution sits 1 floor below that. Neuropod cells plug the gut wall into the highway. Electron microscopy confirmed actual synaptic structures. Optogenetic stimulation confirmed those synapses release glutamate, the brain's fastest excitatory neurotransmitter. The brainstem fires in milliseconds. The wire is real. Now zoom out from that one wire and look at what's on the other end. Coiled inside the walls of your gut, from esophagus to anus, is a nervous system in its own right. More than 500 million neurons, roughly the same number as your entire spinal cord, packed into the meat of your digestive tract. The neuroscientist Michael Gershon at Columbia gave it a name in the late 90s that stuck. He called it the second brain. The technical term is the enteric nervous system, and the Australian neurogastroenterologist John Furness has published the modern textbook account of it. It is built in two layers. Between the outer and inner muscle of your gut sits Auerbach's plexus, which runs peristalsis. Tucked just under the lining sits Meissner's plexus, which runs blood flow and secretion. A complete reflex arc, no skull required. Here is the part that should make you stop. Take an isolated piece of intestine, sever every nerve connecting it to anywhere else, and it still works. Drop food into one end, and the segment produces peristaltic waves on its own. The gut runs itself. About 95% of your body's serotonin is produced not in your head, but in the enterochromaffin cells of your gut lining. So when fear hits, you don't have one brain reacting, you have two. And the one in your gut starts moving first because it doesn't have to wait for any signal to climb a spinal cord. Within seconds of an acute threat, sympathetic nerves release noradrenaline onto enteric neurons and slam the brakes on peristalsis. Circulating adrenaline, already saturating your bloodstream within seconds of the amygdala firing, binds receptors directly on the enteric neurons. The second brain feels the alarm hormonally before the first brain has finished thinking the word. This is the proximate cause of butterflies. A 2023 review traced the sensation to a specific combination: gastric tone drops. Low amplitude contractions ripple through the stomach wall. The sensitivity of mucosal afferents goes up. The signal climbs the vagus to a region called the insula, where your brain assembles a felt sense of what's happening inside your body. The insula renders the whole thing, fairly literally, as the feeling of something fluttering. It's not in your head. It's in your gut, being broadcast to your head in real time. So far, everything has been fast, but there is a second wave, slower and heavier, that arrives behind the electrical one, the hormonal hammer. Inside your hypothalamus, the paraventricular nucleus sits waiting for the amygdala's alarm. The moment that alarm fires, those neurons dump a peptide called CRH, corticotropin-releasing hormone, into a small set of blood vessels running down to the pituitary. The pituitary releases ACTH. ACTH travels to the adrenal glands and triggers the release of cortisol. This is the HPA axis, and the timeline matters. ACTH rises within 2 to 5 minutes of a stressor. Cortisol peaks between 15 and 30 minutes. Adrenaline and noradrenaline are already saturating your blood within seconds. Electrical first, fast chemical second, slow chemical third. While that cascade is launching upstairs, your gut is being shut down by the sympathetic arm. Within seconds of an acute threat, the blood vessels supplying your stomach, intestine, pancreas, and liver constrict. Up to a quarter of your cardiac output can be diverted away from the gut and towards skeletal muscle and brain. That's why your mouth goes dry in a moment of real fear. Salivary glands are downregulated, gastric secretion drops, peristalsis stops. The entire machine of digestion is suspended because digestion is metabolically expensive, and you may need every available calorie to fight or to run. And there is one more thing happening, more quietly, that matters enormously. The CRH upstairs is not only acting on the pituitary. CRH receptors exist on cells in the gut itself. Peripheral CRH increases colonic motility, which is part of why intense fear sometimes produces sudden urgency. It activates mast cells in the gut lining, which release histamine and crank up the sensitivity of local afferents. It downregulates the tight junction proteins that hold your intestinal cells together, briefly increasing intestinal permeability. Hold the timeline. Electrical first, then a fast hormonal flash. Then, the slow, heavy weight of cortisol arriving last. By the time you consciously think the word afraid, the first two waves have already passed through your gut. Here's where the story becomes unsettling. In 2004, two Japanese physiologists named Nobuyuki Sudo and Yasuhiro Koga published a paper in the Journal of Physiology titled Postnatal Microbial Colonization Programs, the Hypothalamic Pituitary Adrenal System for Stress Response in Mice. That paper has been quietly setting the agenda for stress biology ever since. They raised genetically identical mice in two conditions. One group lived in a normal facility, the other was raised in a completely sterile bubble, germ-free, no microbes ever. Then they stressed both groups. The mice with no bacteria mounted a stress response roughly twice as large as the normal mice. ACTH higher, corticosterone higher. Then came the clean test. They gave some germ-free mice one specific bacterium, Bifidobacterium infantis, the kind healthy babies normally acquire from their mothers. The exaggerated stress response calmed down. They tried an enteropathogenic strain of E. coli instead, and the stress response got worse. The most uncomfortable finding came last. The corrective effect only worked if the mice got it early in life. Wait too long, and the hyperreactive stress response was locked in. Past the developmental window, the thermostat couldn't be reset. The set point of the fear system was installed by bacteria during a critical period none of us remember. The team at University College Cork, led by John Cryan and Ted Dinan, built a field on top of this. They coined the term psychobiotics. Specific strains of Bifidobacterium and Lactobacillus can dampen anxiety-like behavior in rodents and normalize stress-induced HPA changes. The effect is usually abolished if you cut the vagus nerve, which tells you the bacteria are talking to the brain through the same wire we discussed earlier. There is also a darker arm involving tryptophan, the amino acid that becomes serotonin. Under stress and inflammation, an enzyme called IDO can divert tryptophan down the kynurenine pathway, producing neuroactive metabolites instead of serotonin. Dysbiosis can quietly starve the brain of serotonin precursor. When you ask whose voice is in the conversation between your gut and your brain, the honest answer is that some of it isn't yours. Trillions of cells that share your body but not your genome are part of the chorus. The human evidence is still thinner than the rodent work, but the foundational claim that gut microbes program the stress response across development is now one of the most replicated findings in neurogastroenterology. The fear thermostat was installed by something other than you. If you find this kind of thing worth knowing, this channel is built for it. Subscribe, and the next one comes to you. Step back. The system as I've described it is elaborate. Evolution doesn't build that much hardware for nothing. So what was all of it for? The honest answer is that this system was perfect once. Picture an ancestral mammal on an open plain. Something moves at the edge of the grass. Within milliseconds, sensory cortex and amygdala have flagged the motion. The vagus is firing, the sympathetic chain is firing, and within a second, the blood vessels in the gut are constricting. About a quarter of cardiac output that was previously going to the slow business of digestion is suddenly available to skeletal muscle. The legs can sprint. Digestion can wait. That's the first reason the gut shuts down. Energy reallocation under predation pressure. The animals that pause their digestion to redirect blood and oxygen to muscle outran the animals that didn't. They left more descendants. We are descended from them. The second reason is harder to see. The gut is the largest immune organ in the body and the densest site of microbial contact on Earth. During a moment of threat, the brain genuinely needs to know what condition the gut is in. Is the animal hungry? Infected? A starved animal should fight differently than a fed one. Vagal afferents feed that information into the decision in milliseconds. Gut feelings, in the most literal sense, are the body voting on how to handle the threat. In 2021, researchers studying wild three-spined stickleback fish showed that anti-predator behavior correlates with gut microbiome composition. The gut-brain calibration of fear is conserved deep into the vertebrate lineage. In the most literal sense, it is older than us. Here's the dark twist. In ancestral conditions, that whole apparatus was engaged for seconds, maybe minutes. The threat was usually physical, nearby, resolved quickly. Then the system reset. The gut got its blood back, the cortisol cleared. Look at the modern threat profile. A late mortgage payment doesn't get resolved in seconds. A hostile manager doesn't get outrun. A push notification at 3 in the morning doesn't trigger a clean fight-or-flight loop with a beginning and an end. The same hardware that was perfectly tuned for a predator is now held in the on position for hours, weeks, sometimes years on end. Cortisol stays elevated. The gut barrier stays compromised. Your gut isn't malfunctioning when you're anxious about a meeting. It's running a perfectly evolved subroutine designed for a leopard. The leopard is now a Slack notification, and the subroutine doesn't know the difference. The clinical world has had to invent new vocabulary for what happens when this ancient system stays engaged for years. Modern gastroenterology no longer calls conditions like irritable bowel syndrome and functional dyspepsia functional disorders. The Rome Foundation renamed them in 2016. They are now called disorders of gut-brain interaction. These conditions aren't all in your head, and they aren't all in your gut. They are dysregulations of the loop. Irritable bowel syndrome is the canonical case. It affects roughly 10 to 15% of the global adult population. Recurrent abdominal pain combined with altered bowel habits with no structural pathology on a colonoscopy. The mechanism is exactly the loop we have been mapping. Visceral hypersensitivity, first of all. The same baseline stretch in the gut you wouldn't notice produces in an IBS patient a felt sensation of pain. Functional MRI shows that when researchers inflate a small balloon in the rectum of an IBS patient, their amygdala and insula light up far more intensely than in matched controls. The gut-to-brain channel is turned up. Underneath that, the same patients show altered serotonin signaling in the gut wall, increased intestinal permeability, and a measurably different microbiome than non-IBS controls. Stacked together, they describe a system whose volume knob has been turned up. The cleanest natural experiment is post-infectious IBS. A meta-analysis found that one acute episode of bacterial gastroenteritis raises the long-term risk of developing IBS by roughly 6-fold, and the risk stays elevated for 2 to 3 years or more. A single hit of gut inflammation can install durable changes in how the central nervous system processes input from that gut. The brain learns the new volume setting and keeps using it. The work of Lucas van Oudenhove at Leuven and Premizel Bursik at McMaster has shown the same loop runs in functional dyspepsia. Patients are hypersensitive to small amounts of fat infused into the duodenum. Their comorbidity with anxiety and depression is well above baseline. The vicious cycle is almost too tidy. Gut symptoms send louder signals up the vagus. The amygdala becomes more reactive. The prefrontal cortex's top-down regulation weakens. Increased anxiety further sensitizes visceral perception. More gut symptoms. The loop literally rewires itself with practice. Hugo Critchley's work at Sussex has mapped how this becomes a felt experience. The insula becomes hyperreactive to bodily signals in chronic anxiety patients. The body becomes a louder broadcaster of itself, and the brain a more sensitive listener at the same time. Nothing in this picture is metaphor. Every step has a named protein, a named cell type, a named pathway. So, if the loop is a real circuit, can the circuit be reached? The answer increasingly is yes, and the entry point is the vagus. Vagus nerve stimulation has been FDA approved for treatment-resistant depression since 2005 and for refractory epilepsy since the late 90s. A small electrode is wrapped around the cervical vagus and connected to a generator under the collarbone, delivering pulses to the same brainstem nucleus the gut talks to. Newer work has shifted toward transcutaneous auricular stimulation. The auricular branch of the vagus runs to the outer ear, so you can stimulate it non-invasively with an electrode that looks like an earbud. Bruno Bonaz in Grenoble has argued this becomes a clinical tool for inflammatory and gut-brain conditions. The immunologist Kevin Tracy at the Feinstein Institute showed in the early 2000s that vagal stimulation has a measurable anti-inflammatory effect. Acetylcholine released from vagal fibers binds receptors on macrophages and quiets their cytokine output. The nerve can talk inflammation down. Behavioral interventions work on the same channel without hardware. Slow diaphragmatic breathing at roughly 6 breaths per minute maximizes heart rate variability. Cold exposure to the face triggers the mammalian dive reflex. Long, slow exhalations do it. Many of the practices meditation traditions stumbled onto are vagal interventions wearing different vocabulary. They worked before anyone could explain why, because the nerve was always there. Psychobiotic work is moving slowly into human clinical trials. The strongest signals are still in rodents. We know the loop is reachable. We are still learning which keys open which locks. The body and the brain were never separate systems. The split between them is a habit of language, not a fact of anatomy. Every flicker of dread, every sense that something is wrong in a room before anyone has said anything, is trillions of cells voting through a nerve that runs straight to the floor of your brain. There is one more axis I haven't mentioned. The heart. The same vagus that runs your gut also runs your heart, and a serious body of work now shows that the rhythm of your heartbeat sensed through cardiac afferents sets a background channel of fear and certainty no one is consciously aware of. Felix Clarer and Cody Bolono and other groups have started peeling apart that signal. What the heart is whispering to the brain, and why some people hear it more loudly than others. That's the next investigation. For now, the next time you feel something in your stomach you can't quite explain, take it seriously. It isn't poetry. It is a signal on a wire in a body whose oldest nervous system has been talking to its newest one for longer than language has existed. Your gut isn't lying to you. It's been telling you something for 500 million years. The hard part is learning to listen. If you want the next one when it lands, subscribe.